have about, I don't know, 15 slides for this metabolic discussion. It is really just skimming the surface. We could spend a whole semester on biochemical pathways. That's what they do in biochemistry. We don't have a whole semester to do that. So this is all about the three main pathways, plus I'm going to tell you about beta oxidation just a little bit, and how we get ATP derived from these fuel sources, lipids and carbohydrates. If we're talking about metabolism, it is the net sum of all processes that build up and processes that break down. Catabolic means we're breaking down, and anabolic means we're building up. So if you actually took fresh plywood and 2x4s and 8x whatever, you can build a brand new house. But we also can demo a building. So that would be anabolic, building up, and this would be an example of catabolic reactions. When we are making anabolic reactions, when we're linking things together like amino acids, we use something called dehydration synthesis. Water is lost when you unite two monomers together, like amino acids making the peptide bond. Water is lost. If you want to break those bonds and get back to breaking everything down to its simplest parts, like in that bottom picture, then you need to bring the water back. Water is our molecular cellular scissor. It comes in and snips everything apart. So that's hydrolysis, when you're using water to break things down. Cells do burn their fuel, but not exactly like we see these logs burning in a fireplace. They are burned in a very distinct stepwise fashion, because otherwise, if we did burn them like this, and by the way, you can burn protein in a fireplace. You can. I'm sure you've done that before. You burned a steak, you can put it in a fireplace and it will burn. It will give off flames. You can burn fats and it will give off flames. We're not giving off flames inside of our body, but we are liberating energy. This is a lot of light energy, for example, and thermo energy. We're liberating energy in different ways. A lot of times students think, well, you need oxygen to burn wood in a fireplace. There needs to be oxygen. Well, that's true, but oxygen is not a fuel. It is not a fuel. If it were a fuel, I would be saving hundreds of dollars every week, and I wouldn't have to feed you all. You would just go, I'm full now. Oxygen is not a fuel, but we still require it. It is the final electron acceptor at the end of these three major processes that I'm going to tell you, and I'll explain what that means. If you have your homework packet out, there's a lot I'm going to be answering. There's a whole metabolism section, and a lot of it also looks like this that you can fill out. There are a few things that I need you to pay attention to. How many hydrogen can you find in a molecule of glucose? How many hydrogens can you find in a fatty acid? And the idea is the more hydrogens there are, the more opportunity to extract that energy, and then we can convert it to cellular energy called ATP. The more hydrogens that you see, the more energy can be liberated. The hydrogen atoms and their electrons, we're going to be basically passing them on to coenzymes or electron carriers, and then ultimately oxygen. Oxygen is going to accept the hydrogen and its electrons, and we're going to turn oxygen ultimately into water, H2O. We're also going to generate ATP. Oxygen and glucose are basically going to be transferred or turned into water and ATP. It's weird to think that we make water through these metabolic processes. We do. It's also weird to think that we're also generating CO2 in the cellular level as a byproduct as well. So the oxygen is going to accept the electrons and the hydrogen ions. More energy rich means the more hydrogen ion atoms that you can extract from a molecule. The most commonly used available fuel for most cells in your body is going to be glucose. And that's where our story will begin, with a process called glycolysis. We can burn fats, and fats actually give us more ATP because they are long hydrocarbon chains. I'll show you a picture of that towards the end of this PowerPoint. We can use protein as a fuel, though most of the proteins in our body are considered to be structural, meaning we need them, and also functional, meaning they, like in skeletal muscle, they allow for muscle contraction, movement. So that's like a last-ditch effort. We really don't want to burn the proteins in our body for fuel. We're just going to have to make them again. And there's usually a consequence, like albumin levels can go down, skeletal muscle mass goes down. It's not a good thing to do. We also could, in theory, burn nucleic acids. We could. However, that is absolutely not a good idea, because if you start burning your DNA as fuel, then you're losing the recipe book to make proteins, right? That's not a good thing to do at all. So that's not really an option, but it is possible. Just under normal physiologic conditions, we don't do that. Now, when I say all roads lead to mitochondria, that means ultimately, after processing glucose, or using fatty acids, or burning proteins, which is we can and we will, but it's usually the last-ditch effort, all roads will lead to the mitochondria. All roads will. And that is going to be an important concept that I'm going to talk about this entry point into a metabolic pathway called Krebs cycle, also known as the TCA cycle, tricyclic acid cycle. So there's going to be a rate limiting factor that I want you to pay attention to. And we were talking about carbohydrates earlier with diabetes. And I told you what a serving of a carbohydrate usually looks like for bread and rice. I know, he's like, But I am tired of people saying, oh, carbs are bad. Carbs are bad. Carbs are bad. I'm on a low carb diet. Okay. Anytime you put yourself on some sort of diet, let me tell you why they usually work. It's because you're being mindful of what you're putting in your pie hole. You're paying attention to it. You said, I was supposed to be good today, meaning eating more healthier choices. You didn't do badly. Starving yourself is also not good. And carbs are required. They are necessary. You cannot burn fats without carbs. Period. End of story. And again, people who are on a low carb diet, it does work for them. True. You want to know why? Want to know why? Want to know why? One, they're mindful of how much they're eating, and they are probably now getting back down into what should be a normal serving. It's normal servings. Most of us eat well beyond a typical serving for a carb. So It's not necessarily they're on a low carb diet. They're actually now being quite more responsible with their serving size. But then you have people that take it a little too far and they say, well, if I lost all of this weight by watching how many carbs I eat, imagine if I cut it out altogether. And that is not a healthy situation. It's not a winning situation at all. You require carbohydrates in your diet. You can't burn fat without them. We say fats burn in the flame of carbohydrates. You must have them. The idea is portion management. So let's start our talk with glycolysis. Now, on the next slide, I'm going to show you all the steps of glycolysis. I don't want you to memorize every step. If you were in biochemistry, you would be mandated to do that. That would be part of the outcomes that they would ask you to know. Not in this class. I'll be happy if you can tell me where does it happen, how many ATP are produced, and how many coenzymes are we sending to the mitochondria. That's what I want you to know. Where is it? What do we start with? What does the starting material look like? And what do we finish with? That's basically what I'm asking. So if you look at the board, I have a little project that I've written up here. And we're going to go through this step by step and try and fill in these blanks. I should also add where. So let's start with that. Glycolysis is going to be in the cytoplasm. And because it's in the cytoplasm and does not directly involve going into the mitochondria, we say that this is an anaerobic process. You don't need oxygen for this process to occur. What do we start with? We start with a 6-carbon chain. Glucose has 6 carbons, C6, H12O6. So how many hydrogens are in there? C6H12O6. That would be 12. That's right. Good job. So that means there's good opportunity for us to cleave off these hydrogen ions and their electrons. But I want you to know something. These coenzymes that I'm telling you about, specifically NADH and FADH2, I want you to think of them as oven mitts. When do you use oven mitts? To take something out of the oven that's very hot. I want you to think of hydrogen and its electron as a hot potato, really, really hot. And we got to do this step by step, I told you, because otherwise they release a lot of energy very, very quickly. So you can actually put substances in a calorie bomb. That's usually what we call them. And you literally light it on fire and it will tell you how much energy is released. So these hydrogen and electrons, hot potato, hot potato, hot potato, and we got to transfer them to the mitochondria. Hot potato, and the coenzymes I should say, are NADH and FADH2. They're the oven mitts that say, we got you, we're going to take you to oxygen. It's okay, hurry up, let's go. Be mindful of how many of each of those are made in each step or process that I'm going to tell you about. If you don't have oxygen at all, the glucose, like I said, can be burned. You will generate some ATP, and you will generate some of these coenzymes that I was telling you about. But it can't go past that And now you're going to be forming lactic acid. And the only way to get back to pyruvate, and I'll explain what pyruvate is if the lactic acid goes to the liver and goes through a process called the Cori cycle. So you can't, it doesn't give you a lot of energy. If you're at the gym and you're on the treadmill running so fast, or you're in a cycling class pedaling so fast, or you're in an aerobics class, whatever workout you're doing, if you cannot talk without going, then you're probably going above your oxygen availability. You're going to go into anaerobic metabolism, and your muscles are going to be very, very sore. So you want to make sure you can at least have a sentence that you can speak to someone else in the class. That does not mean, for the sake of everyone else in that gym, it does not mean you carry full conversations with the person next to you on the treadmill. That is not what it means. If that's what you want to do, walk to a coffee shop, get a coffee, and then walk back. I don't want to hear about when was the last time you waxed your legs. I don't really need to hear that. I think they should have a visiting room. You know how they have a nursery for the little kids? Have like a visiting coffee room for the adults that just want to talk. Okay, glycolysis. We start with a 6-carbon chain called glucose. It has 12 hydrogen. And the first thing we have to do is invest some cellular energy. We have to invest some ATP. And we invest 2 of them. 2 ATP. Then we're going to start ripping apart this 6-carbon molecule into 3 carbon chains. And we're going to make 2 of them, right? 6 cut in half is 3, and you get 2 of them. So as we start cutting, look at here, we generate 2 coenzymes. Right there, we've got 2 oven mitts with the hot potato. So you might want to put here, for under your glycolysis, number of coenzymes, 2. Then we get the payoff phases. As we continue to rip and rearrange these phosphate groups and take them off, we actually make back 4 ATP. 4 ATP. 4 were made total, but what was the net gain? 2. If we had to invest 2 and we get 4 back, we're still up by 2, but we don't have all 4. We had to invest some. Number of coenzymes, 2 NADH. End product. At the end, we have 2 3-carbon chains, and that is called pyruvate, or pyruvic acid. So that is next. 2 pyruvate. And they are a 3-carbon chain. And these pyruvate molecules are going to feed into the TCA, the tricyclic acid cycle. It occurs in the mitochondria. These last 2 steps occur in the mitochondria. We're not going to say how many total ATP are used for these 2 steps because we're going to focus on the net ATP. There isn't going to be an investment phase for either one of these, so that does not apply. So let's go to the TCA. It happens in the mitochondria. Anything that happens in the mitochondria, we need oxygen to be present. The tricyclic acid cycle does not directly use oxygen, but in order for the pyruvate to funnel to the mitochondrion, we have to have oxygen available. You do not need to memorize all the steps for the TCA cycle or Krebs cycle. You do not. You do not. But there are some things you do need to know. First of all, pyruvic acid, pyruvate, cannot directly go into this cycle. Up at the top, you can see what I refer to as the preparatory phase. And by that I mean we have to take the 3-carbon pyruvate and cut off a carbon. Now we will have a 2-carbon molecule. That 2-carbon molecule will bind to a 4-carbon molecule to give us a 6-carbon molecule. That 4-carbon molecule right here, oxaloacetate. Oxaloacetate. And then we're going to have a 6-carbon molecule that is made. 1, 2, 3, 4, 5, 6. So 2 carbons attached to the oxaloacetate. And then as that 6-carbon molecule goes around the merry-go-round, as I like to call it, you have 1 carbon removed, and then another carbon with hydrogen ions also removed, and you restore the oxaloacetate. Then the next pyruvate, because remember we get 2 of them from glycolysis, will now join to oxaloacetate. As it goes around, 1 carbon is removed, then another carbon, and you recreate oxaloacetate. What do we gain when we go through the Krebs cycle, including the prep phase? For every pyruvic acid that enters, we get 1, 2, 3, 4 NADH. For every pyruvate. Be very careful with the wording in your homework packet. It would be a different answer if I asked for every glucose, because then it would double, because you get 2 turns. 1 pyruvate goes, then another one. Be mindful of the wording. So now, is this my good one or my bad one? So now we have 4 NADH. And that includes 1 that's made by the prep phase, preparatory phase, as I like to call it. What else do we make? We make 1 FADH2. But if we were considering the 2 rounds, it would be 2 total. Okay? So I'm going to put 4 NADH, but we get 2 turns, so it's really 8 total. We make 1 FADH2 for every pyruvate, but because 2 of them go through the cycle, that's technically 2 total. Okay? Can you follow that? Where does this happen? In the mitochondria. For 1 spin, you get 4 NADH. For 1 spin, 1 FADH2. But we have 2 pyruvate coming through because we ripped glucose in half. That's what we did. That's where the numbers are coming from. Okay, now we gotta turn in, and I believe this website is still available. It is through the National Institute of Health, I think. Anyway, it's funded by the National Science Foundation. It's a pretty good video. It's very simple to follow. Let me tell you about these coenzymes and what I mean by their cash out value. When my sons were young, they loved going to Chuck E. Cheese. And I don't know if you've ever been to Chuck E. Cheese, but here's how it works. You enter Chuck E. Cheese with these children, And I firmly believe that Chuck E. Cheese is turning these children into future gamblers that will have a problem. Here's why. You bring good, hard-earned cash. You put it into a machine, and the kid has a cup, and out comes, like in a slot machine, clink, 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 tokens. Worthless money. But not to them. They think they're rich because they're even colored gold. So then these children go around putting these coins, worthless coins, into the machines, and depending on how they perform, they outspews tickets. And some of the machines, again, like a slot machine, future gamblers. Kid just puts in the coin, and there are these little rakes that just move back and forth and back and forth, and the child is hoping that eventually it'll push over a whole bunch of tokens, and then they'll get a longer strand of tickets. So then they run out of their coins. Mom and Dad say, enough. No, you're not getting another $20 from me for this. And the children in Chuck E. Cheese, they love to get long strands of these tickets, and they parade around like they have robes of tickets, and they're just so proud. And every kid is trying to be like, how long is the train, right? How long? And then Mom and Dad say, okay, it's time to turn in your tickets and pick out your prize. And we got to go. You've got 5 minutes. Well, then these long tickets, you can feed, it's called the munch machine. That's what it's called. And you can feed in these long strands of tickets, and it'll just keep dragging them in, right? And it makes annoying sounds. There's like 5 of them around the whole facility. And all you hear is but my kids, because they didn't want to leave, they would tear off one ticket at a time to feed it into the machine one by one. And then when they were done, they'd hit the stop button, and out from the munch machine would come out a white piece of paper that said how many tickets they had. Again, what was the exchange? We went from real money to gold coins to tickets that were long strands to a piece of paper, and then they take it up to the counter, the exchange counter for toys, right? And they exchange this little piece of paper, and they can pick out some candies, they can pick out toys, whatever. They can even save these tickets and get a whole bunch of them and get bigger prices, right? So my boys would pick out their toys and maybe some candies, and those toys would be broken by the time we got to the parking lot and in the car. They were craptacular toys. Excuse my language. So instead of the toy counter where you exchange tickets, I want you to think of these NADH and FADH2 coenzymes as the tickets. And we're going to exchange them for ATP, the electron transport chain. It works well for exchange toys, right? Counter, ETC, and with my Chuck E. Cheese analogy. So that's what I mean by cashing out. Now, this is the electron transport chain. It has quite a few proteins that are involved. And I just want you to notice that at the beginning, here's complex 1, 2, 3, 4, and then finally ATP synthase. So 5 proteins. NADH goes in early, goes in at the beginning of this electron transport chain. Because it goes in first at the beginning, it cashes out for 2.5 moles of ATP. for every NADH, 2.5. FADH2 comes in not at the beginning, roughly second in line. And because it comes in later in the chain, it does not cash out for very much, 1.5 moles of ATP. Now, a lot of times professors will say 2.5 ATP, and I remember one of the chemistry professors, I said it, and he said, how do you get 2.5 molecules of ATP? I said, you don't. It's for moles. Oh, well, then that you can have. I'm like, thank you very much for confirming that. Now, a lot of your textbooks, I'm sure, maybe you've learned this before in high school. You learned that NADH cashes out for 3 ATP, and FADH2 cashed out for 2. I want you to know that it's an average that I'm giving you. What they put in the book is not incorrect. It is correct. I am giving you an average, and I'll explain it to you this way. Do you know when a leopard or a lioness is most successful when it's hunting its prey? Do you know when? When is it most successful? When it's really hungry. When it's really hungry, it is most successful. Its life is literally on the line. And studies actually show that for even people who participate in athletic sports, they do better if they don't have like a full breakfast on their stomach, that they eat a very light meal, making them hungry to play, right? They're more energized. I know it sounds like it should be reversed, but they're sluggish if their tummies are full, especially running in a soccer game. You don't want that. So I used to starve my children before their soccer games to make them hungry for that ball. I didn't starve them, but I was mindful of that. It does equate to our human lives if we're competing in some level, just like a lioness or a leopard is competing for that prey. Okay, so my point is, when cells are really desperate for fuel and really desperate for more ATP, they become more efficient, like skeletal muscle. And then there is a bigger cash out value. But in other cells, not so much. I gave you the average. Because the numbers I'm going to show you on the next slide, you're going to say, that's not what I learned when I learned about glycolysis and Krebs cycle and the electron transport chain. I remember we got more ATP. Again, I'm giving you an average. Okay. Now there's a caveat here though. These two NADH that were made way out in the cytoplasm, they have to cross the mitochondrial membranes, plural. And when they cross the mitochondrial membranes, they lose some of their energy. And so these two will cash out at 1.5, not the full 2.5. Get lower energy value because they came from the cytoplasm. So let's tally this all together. And keep track of our overall net ATP based on my numbers, giving us an average. In glycolysis, we had an investment of 4 ATP, we make 4 ATP, but we had an investment of 2, and we have a net gain of 2, all right, so that's 2. Then we go into the TCA cycle and electron transport chain. I forgot to write down in the TCA cycle, for every spin, we get 1 ATP. But remember, we have 2 spins So it's really a total of 2. Okay, so we have these 2 that are made right here in Krebs cycle. It says substrate level phosphorylation. And it looks like, no, Kara, we're not making ATP. We make GTP, yes, but then that is converted into ATP. Now we need to cash out our NADH molecules, coenzymes, that were made inside the mitochondria. How many did we make total? We have 8 total that are made because we have the 4 for every spin, including the preparatory. 8 times 2.5 is 8 times 2 is 16. Good. And half of 8 is, so 16 plus 4 is 20. Okay, good. We make a total of 2 FADH2s. They cash out for 1.5 each, so we get a total of 3 ATP. Why do I have 3 ATP up here for NADH? Those are the 2 that were made up here. They have to cross the membrane. They cash out for 1.5 each, giving us 3 ATP. If we add 2 plus 3, that's 5, plus 20, that's 25, and 5 more, we get 30 ATP. You might have learned different numbers, like 34, 36. Again, that depends on which tissue your previous professor was drawing from. I'm giving you the middle of the road. Your past professor was not wrong, and I'm not wrong. I'm just explaining why I'm giving you different numbers. Which numbers do you think you should use for testing purposes on my exam? Mine. Good. Okay, so we're in agreement. And then, of course, down here at the very bottom, you're seeing that the oxygen is the final electron acceptor. It accepts the electrons and the hydrogen atoms, and we turn it into water. We also generate CO2 as a byproduct of the Krebs cycle, and not really CO2 from the electron transport chain, but this is the preparatory phase and then the actual cycle itself. In your homework packet, it asks you what is oxidation and what is reduction. Oxidation is we are removing an electron. And reduction is we're giving that electron to something else. We burn our glucose fuel, we are oxidizing it. We are extracting the electrons from it. When we give those electrons to oxygen, we are reducing oxygen. We're also including the hydrogen atoms, and we get water. Lipids come in a variety of forms. I'm going to tell you about fatty acids and glycerides and how we metabolize them. I'm going to show you the overall process. Do you think I want you to know the individual steps? That's right. No, I don't. And I can't really give you an exact amount of ATP that is generated by burning a fatty acid, and I'll explain why in just a moment. A fatty acid is a long chain of hydrocarbons. Look at all the hydrogen. More hot potatoes means more coenzymes, means a lot of cash out. I can't tell you a definitive amount for ATP because fatty acids, the chains of hydrocarbons, come in different lengths. You can have a 20 carbon, you can have a 22 carbon, different lengths. So I'm going to show you for a traditional 20 carbon length of a fatty acid. When we cut the fatty acid, we cut off 2 carbons at a time. 2 at a time. What was the 2 carbon chain that we converted pyruvate into in the preparatory phase? It was acetyl-CoA. Remember when I was doing this? A 2 carbon chain meets up with a 4. That 2 carbon chain was acetyl-CoA. And as we go through the Krebs cycle, 2 carbons are removed and we recreate oxaloacetate. Same thing with burning a fatty acid. 2 carbons at a time, instant acetyl-CoA. Glycerides are a glycerol backbone, shown here, a 3 carbon chain, but then attached to an oxygen, we have dehydration synthesis, and now we have a triglyceride, 3 long fatty acid chains. We can burn these triglycerides, 1 fatty acid chain at a time, and generate a lot of ATP by burning them. We get more ATP for burning fat, but here's the cruel reality. In order to burn your fat, you need to work out at least for 20 minutes. If your adipocytes would talk to you, they would say, wait a minute, let's not release the fatty acids yet. Let's see if he's serious about this weight loss thing. 20 minutes, and you have to make sure you are in an aerobic phase. If you are exerting yourself beyond 80%, you are not getting enough oxygen to be a final electron acceptor. The sweet spot is actually around 60% for most people. But nothing more than 80%, not recommended. We use triglycerides not just for energy source, but insulation and protection. When we cut the lipids, like the triglycerides, we are using lipolysis. And when we use lipolysis, we're liberating those free fatty acids, and then they can be broken down in a process called beta oxidation. When we have this breakdown of the fatty acids into 2 carbon fragments, acetyl-CoA, they then can enter the TCA cycle. For every turn of the TCA cycle, we generate an ATP, we generate 3 NADH and 1 FADH2, and they are going to cash out for a lot of value. But again, you can't provide a lot of energy in a short amount of time. Lipids don't like to go into the bloodstream and then go to other parts of the body to be used as fuel. 20 minutes or longer. Now these are the steps for beta oxidation. There are a lot of in-between steps, and here's what I want you to know. Where does it occur? It begins in the cytoplasm and finishes in the mitochondria. The acetyl-CoA go in through Krebs cycle, and then our coenzymes go and cash out at the electron transport chain. Here is a 16 carbon fatty acid that started as an 18 carbon. We cut off 2 carbons at a time. A typical 20 carbon fatty acid can generate over 100 ATP, approximately 144. That's a lot. But again, it takes a while to mobilize those fats. Now here comes the punchline with me telling you carbs are necessary. They are absolutely necessary. Remember that 4 carbon chain that begins the Krebs cycle, oxaloacetate? It's carbohydrate derived. And if you are on a low diet of carbs, that oxaloacetate can leave the mitochondrion, go back into the cytoplasm, and be used in an anaerobic process to make ATP. which means your entry point to the Krebs cycle. And remember, all roads lead to the mitochondrion. You now have no entry point. You don't. You've lost it. And that means you're not burning your fats completely. You're not burning your fuels completely. You have a roadblock. This can lead to ketosis and ketoacidosis. What's the difference? Ketosis is what you experience in the morning when you wake up. What aren't you doing when you're sleeping? What aren't you doing? Eating, okay? And probably a lot of other things, but what aren't you doing? That's a big one. If you aren't eating, then you start metabolizing alternative fuels, like fats, and maybe even some proteins. And you wake up in the morning and you have stinky breath, don't you? You've got morning mouth. Well, the stinkier, the better. It means you've been burning other fuels. So now I want you to wake up tomorrow morning and I want you to go to your loved ones or your pets and go, "Good morning. How does my breath smell?" And they're going to go, go brush your teeth. And you're going to say, "But I've been burning fats all night long." Arguably a reason to sleep more. Yes? Arguably a reason. And actually studies show that people who do sleep a good 8 hours, they actually tend to be thinner. I know. If you don't have that oxaloacetate, you can tip into ketoacidosis. That's what happens with people who really, really limit their carbs. You can't burn everything completely. You build up these ketone bodies, and they are called acetone, that's volatile. It's a ketone body. But these two are not only ketone bodies, they are keto acids, and they only come out in your urine. The acetone comes out in your breath. That's the morning milk. Acetoacetic acid and beta-hydroxybutyrate are the other two. They are acids, and it will make your blood pH drop. When we get to Unit 4, I will be teaching you about acid-base balance. This is lethal, absolutely lethal if it's not controlled. So are carbs bad? Are they necessary? Is it about portion control and understanding what a serving is? There you go. Have a good night. I'll have a treat for you on Wednesday.